Bonjour à tous. Cette conférence a lieu en anglais. J'espère que ça convient à tout le monde. Oui? Super. J'ai le plaisir d'accueillir Tom Battin. Professor Tom Battin, you're the head of the River Laboratory. And with your team, you study glaciers, which is not just a question of water flooding and melting, but it's also a question of ecosystems, chemistry, biology, physics. And today you're going to focus on the rich life that comes with glaciers and that might disappear when glaciers disappear. I have to say that you just received the Vernadsky Medal, which is a prestigious award from the European Geophysical Union. Tom, the floor is yours.
Thank you very much. Merci beaucoup. Two words in French. Vraiment merci pour s'avoir égaré ici. Je sais que ce n'est pas la meilleure heure de la journée et j'essaie d'être court, comme ça vous allez faire le marathon où vous allez manger. Je vais faire ça en français, c'est plus facile pour moi. S'il y a des questions après en français ou en allemand, ou même en luxembourgeois, on y va. So, welcome to this talk, Cruising the Planet's Roof, to disclose the unseen. And this has been and still is a fantastic project that has been most generously funded by the Nomis Foundation. Now, let's talk about the unseen. It took me quite a while to prepare this slide here specifically. So I guess we all are aware what I mean by the unseen. So this is the unseen, or part of it. We're never alone. We have little companions, unseen yet, millions of them on our skin, in our intestine, etc. Right? Soon, some of you are going to go for a swim in a lake or in a larger river. And whenever you get a couple of milliliters of water in your mouth, you have a mouthful of unseens. So this is really the unseen majority. The unseen majority I'm talking about today is these one here, microorganisms. And this is just to give you a kind of a feeling what we're talking about. So here we have like the kind of the scales. So we are somewhere here, like 1 meter, some of us are almost 2 meters in here. And the microbes, so here we're talking the order of a micrometer, so thousands of a millimeter. And the microbes that you would most likely swallow in the lake, they are half a micrometer, they are even not an entire micrometer. So we're talking about extremely small microorganisms. We don't see them, and yet they are very, very abundant and numerous. Since we don't see them, so we can make them visible using very special microscopy techniques. So here on this lower panel, you see a bunch of bacteria hanging out together. This one here is quite spectacular. So this is a microbial or bacterial cell, and that has many, many little friends around. These are viruses, or we call them phages, right? And these phages, they really rely on bacterial hosts to kind of reproduce. So since these bacteria, we cannot see them with our naked eye, what we do need to do is we sequence their DNA, for instance, right? And once we have the sequences of the DNA of these bacteria, to some extent, we can understand who they are, how they are related to each other, and also to some extent what they do potentially. Now, bacteria rarely come alone. They like to kind of aggregate, and they like to form what we call biofilms. I'm going to tell you a little bit more on biofilms in a second. This here is the beauty of a biofilm, right? So this here, what do we see here? These are kind of like fungi, and then like in green, some what we call cyanobacteria, and then here colors, so these are false colors, various microorganisms. And they all live together in what we call microbial biofilms. So these biofilms, they are true microbial megacities. Life in these biofilms is truly crowded. We find in a cubic centimeter of such a biofilm, for instance, millions and millions of cells. We do find in there not only millions and millions of cells, we also find thousands of different species all coexisting together. That's why we call them megacities. There's lots of immigration going on in these biofilms. There's also lots of emigration going on in these biofilms. At the same time, there's death and there's birth. So it's exactly what happens in a city as well. Within these biofilms, there can be lots of cooperation, even share of labor. Some of these microbes, they know they can talk to each other. It's not only that they like to cooperate, but there's also a very high degree of chemical warfare. So it's not only about friends, it's also about foes. These biofilms, they have been around on this planet since quite a while. Since more than 3 billion years, biofilms have kind of dominated the life on this planet. There is the idea also that biofilms may be kind of first forms of multicellular life. For those of you who like diving or snorkeling in warm waters, you may have encountered stromatolites. These are kind of biofilms, and stromatolites have been around in the ancient times of our Earth's history. So why are microorganisms important? Microorganisms are like the conductors of the major biogeochemical cycles on this planet. So that's number one, right? So whenever you want to have water clean in a river or in a lake, it's the microbes that do the job. Whenever you want to have a soil fertile, the microbes do the job. Whenever you want to have a compost in your garden, the microbes do the job. Again, it's the unseen majority and yet super important. We don't see the microbes, yet they are super diverse, and the diversity or the biodiversity that we see, from plants to elephants and lions, they all rely on the invisible biodiversity of the microbes. This is just to give you an appreciation of what the biofilm in a river may look like. So it's a kind of a slimy, a slimy mega city, and you have like these little animals in here. So these are like the cows. They like to graze on they are grazers. So they feed basically on the microbial lawn, etc. These microbial biofilms, they are true microbial jungles. And this is not a joke, this is serious. So within these biofilms, in streams and rivers, you find representatives from all 3 domains of the tree of life, as you find in the jungle. Well, we don't have any chimpanzee in here, but we have lots of what we call metazoans, etc., and we are part of the metazoans as well. So they are highly, highly, highly diverse. Why do we study this kind of microbial biofilms in streams? So today we understand that streams and rivers, they form beautiful networks. So this is the river network of Spain. So you can read the shape of Spain from this simple map here. And river networks, they kind of integrate and perfuse our landscape. Today we understand that these river networks, they are extremely important for the global carbon cycle. We want to understand the global carbon cycle well, because we have been disturbing the global carbon cycle tremendously over the last 150 years and even longer, with major consequences for the climate, as we all know, hopefully by now. Just to give you a kind of a number, so the worldwide streams and rivers, they outgas into the atmosphere per year 2.3 billion tons of carbon dioxide. This is the equivalent flux as the world's ocean draw down from the atmosphere per year. So now, this outgassing flux here, please do not panic. This is a natural flux. It has nothing to do with us. It's a natural flux, right? But this is kind of cool because like the river and stream networks, they contribute at most 0.5% by spatial extent to the non-glaciated land surface of this planet. And yet they are so important for the global carbon cycle. And some of these secrets lie in the fantastic form of the biophil mode of life. So let me take you now to the roof of our planet and the biofilms that live well hidden in these water towers. So we call the high mountains, like our Alps here, for instance, or the Himalayan, these are the water towers of this planet, as we can see here. So this is probably the Mount Everest, I guess, or this one here. Anyway, this is the Himalayan here in the background, and these are the big, big rivers that drain down the Himalayan into India. So these are the water towers. And the water towers are melting away before our eyes because the climate is warming. This here is a very nice representation of the increasing annual air temperature starting in 1880. And then, as you can see, the color is changing. And then here from the 80s onwards, it's getting really, really red. So this is in degree Fahrenheit. I'm sorry for this. But we understand that we kind of overshooted the climate as it was at least before Second World War. And it's because of this, there have been some predictions made recently. By the end of the century, we may lose something between 50 and 80% of the world's mountain glaciers by the end of this century, right? And this is an unprecedented environmental change. And let's not forget there's all these hundreds of thousands of streams and rivers that are being fed by the mountain glaciers. They provide us very important ecosystem services. Some of these ecosystem services, they are like supporting, as for instance, like water resources. They are regulating, such as the buffering against droughts. They are also provisioning, such as food and sanitation, or they are simply like cultural, such as recreation and spirituality. So we are losing this, right? And so we were asking, what else are we losing? And so we are very much interested, as you may have noticed by now, in this microbial life. And today, the microbial life in these streams that literally drain the roof of our planet is way less understood than the microbial life in the deepest and the remotest ocean worldwide, despite the fact that these streams, they are so vulnerable to climate change. Whenever you have the chance to go to a glacier, just be careful. There are rocks and stones falling down all the time, sometimes even chunks of ice, so be careful, but it's a fantastic system. So here's the stream that comes out. This is my friend Mike Stiller's. These streams, they are really, really extreme ecosystems. It's, as the ice is melting, it's freezing cold. During winter, it's dark. Everything is covered below ice and snow. In summer, the UV radiation, as we all know, when we go up there, is pretty high. These streams are extremely turbulent, so it's not fun to live in there. Plus, the sediments are highly unstable, and there's not much to eat in these streams. So we were curious to understand the microbial life in these streams, and over 4 years, we conducted expeditions throughout the world, even during the pandemics, and we started here in New Zealand, and then it took us all the way here through Central Asia, the Himalayan and the Russian Caucasus, for instance, Scandinavia, Greenland, the European Alps, of course, and then also we went to Ecuador, to the Chilean Andes, and then the last expedition took us to Alaska. We had to travel with the seasons to optimize our time, and so we kind of went to the southern and to the northern hemisphere as the seasonality and the whatever snow, etc. conditions allowed. So in total, we had something like 12 expeditions here. So my 4 team members and dear friends and colleagues, overall, they walked. They literally walked something like 1,350 kilometers, and they climbed in altitude more than 80 kilometers to really get to these glaciers. They brought home more than 16,000 different samples that we all analyzed here at EPFL. So going on this kind of expeditions, it's lots of fun. It's extremely heavy work. So you have to prepare yourself well. So we all did survival training in the snow, and then we all got not only like vaccinated against COVID, but everything else that is around. And then, you know, we brought home the thousands and thousands of samples, so you better prepare yourself well. And then, whenever we went to a country, we needed various permissions, etc. So we really got through the bureaucratic machineries of some of the most beautiful countries on this planet. Just to give you an idea, when we went to New Zealand, we required a special permission from each Maori tribe that we visited, basically. It was lots of fun. We learned a lot. And here you see, every time we traveled, that's the way we traveled. We sent via cargo almost like 400 kilograms of material, weeks before we actually left, etc. And once at the spot, you know, we went through the misery of bureaucracy and bribery, etc., etc., etc. These are some pictures here. You see this one here, here, and here you see it again. So we'd always traveled with liquid nitrogen in so-called dewars, special containers, where the temperature is between -80 and -100 degrees C. So that's basically our portable super deep freezer to kind of preserve the samples at the spot. And the art is really to never ever break that temperature chain when you bring back the samples from the roof of this planet to EPFL. This is just to give you a taste. This was one small expedition in Nepal. So here we had 714 kilograms of food and some of our equipment, and here we required something like 30 porters and Sherpas and guides to take us to the spot. So here we are packing everything into this funny, lovely bus, and again our duo with the liquid nitrogen. And so this is just to give you, I hope this works, a little impression of, so by land, so how do we approach these glaciers? So we took advantage of everything that we found, you know, all these lovely roads. This was in Greenland by the sea, excuse me. This was like in New Zealand, amongst others. There we had to rely quite a bit on helicopters, almost unfortunately, I have to say, but some of these glaciers are extremely, extremely remote and really, really hard to kind of approach. This year was in Nepal, so you walk up the hill for one entire week before you even see a white mountain. And I think this year was somewhere in Caucasus or in Kyrgyzstan. So here is Matteo quite happy on that horse. Then we had like a kind of a base camp, and so there were different types of base camps. So this is a base camp, or sometimes it was like in what we call a lodge, for instance. And life in these base camps, you know, there are plenty of like surprises. You wake up in the morning, there's snowfall, and yet you have to work in the snow. Or as you can see here, it's freezing cold even in there. And we use these base camps also as kind of labs to prepare and preserve the samples. And that's how we worked really like at the snout of the glacier where we kind of took the samples. And I really, really have to acknowledge, and I can't acknowledge it enough, the help of all our local friends, being like in Nepal or like in Africa. I mean, they were just absolutely great people. Without them, all this work would definitely not have been possible. So let me guide you rapidly through a couple of pictures so that you get a better understanding of what these biofilms look like. This is like a beautiful piece of art, at least to me. Well, I'm a little bit biased, but this is a fantastic form of life, as this one here. And then during winter, when there's no more water, so we are not aware of the fact that water is really limiting in these ecosystems, right? So during winter, these biofilms, as you can see here, they literally freeze dry. And then we don't know whether they are able to kind of resume life after the winter. We don't know yet. So I'm gonna expose you now to a couple of our results, just a few of them. So overall, we found something like 300,000 bacterial species in these glacier-fed streams. So there's a very, very, very high diversity in there. So per glacier fed stream, we found on average something like 2,800 of these bacterial species. So for our analysis, we retained out of these one only 1/6, so something like 50,000 species. Otherwise, we would not have been able to do all the complex bioinformatics. So this is just an example. So that's the way we know our the fauna on our planet, right? So we understand that there are some elephants in Africa, there are also some elephants in India, for instance. There is like the ice bear is on the North Pole, the ice bear is not on the South Pole. And so we call the distribution of animals and the distribution of plants, that's what we call the biogeography. And up to now, we were not really able to tell whether there's such a biogeography for microbes, and yet there is. And that's something that we see here, so just forget, I mean, they are nice color. So each dot here is one bacterial community from one glacier-fed stream. And just what I want to show you here, there's a clear divide between the glacier-fed streams, or the microbes in the glacier-fed streams, between the northern hemisphere and the southern hemisphere. So yes, indeed, we were able to show that there is a kind of a biogeography of these microbes as well. What is kind of cool is that more than half of the 50,000 bacterial species that we retain for our analysis, they are endemic. What does that endemic mean? More than half occur in one single mountain range only. Like, for instance, New Zealand is very well known for endemic plants and animals. Up to 60% of the bacterial species in New Zealand, we found them in New Zealand only, and only in New Zealand, right? And this is something, it's really unusual and extraordinary, and this is possibly, when you look at this, these glaciers and their streams, they're extremely isolated and deeply incised, so it's probably because of this spatial isolation that we have this high degree of endemism. So this is bad news. Why is it bad news? As the glaciers in New Zealand shrink and disappear, there may be a high likelihood that these microbes disappear as well because they occur in New Zealand only. Right? It's the same story for Ecuador, etc. And yet, we have some suspicions that nature has kind of evolved a trick to mitigate this kind of climate change impacts. Well, we can discuss about this later if you are interested in this. Now, on the contrary, there's only like 0.5% of all these bacterial species that are what we call cosmopolitans. They like to travel around the globe, and wherever you take a sample from a stream, you find them in there, right? So these are the ones that are extremely well adapted to the overall conditions in these streams. We did a lot what we call metagenomics, and so the metagenomics has unraveled some of the secrets that make the life in the microbial life in these streams so successful. So if you want to do well in these streams, be a generalist, don't be a specialist. Try to have a high level of functional redundancy and diversify your energy acquisition strategy. So for instance, to acquire the energy from the light, we found, and this is kind of really intriguing, you develop different acquisition strategy, you better grow fast, and you recycle your nutrients as much as possible because there are very little nutrients in there. They are really nutrient limited. So we could learn a lot from these fantastic communities. And then we're looking into the future and we're asking the question, so what does the future of these streams look like? And so the take home here is that the future is green. So these streams, they will turn into green. That's because like the water are becoming clearer, and as the water are becoming clearer, there's more light available. As there's more light available, the primary producers, so the forest of the microbial jungle will grow. And this relieves this microbiome from energy and resource limitations. And yet we don't really understand what the consequences for the carbon cycling is. So this is food for the future. 2 more slides, then I'm done. So the take-home messages for you here, if I may say so, and then also some of the next steps. So we understand that the glaciers are melting worldwide. We also understand that we are losing massive water resources, quite important for millions and millions of human populations living at the foot of these big hills. And at the same time, the sea level is rising. Right? So we were asking the question, what else are we losing? And to really be able to ask this question, so today we are moving what we call towards like global river science. We need to have a global overview. That's why we did all these expeditions. So we're also losing microbial diversity quite a bit probably. And the ecosystem properties and the related biogeochemical fluxes of the world's highest streams is changing profoundly, and yet we don't understand what the downstream impacts are. So questions that we're going to ask over the next couple of years is, what is the genetic repertoire? As we are losing the biodiversity, we are losing lots of genes, and yet we even don't know the functions of these genes. What we're going to do is some other people is we do bioprospections for novel antibiotics, for instance, and we try to reconstruct the life of the ancestor that lived in these ecosystems. So this has been a very complex and is an interdisciplinary project, and so here you see some of the disciplines that are involved in this, with this project. I am having the absolute great privilege to be able to work with a wonderful team of scientists and PhD students and technicians here at EPFL, and then we enjoyed a lovely cooperation with the University of Luxembourg, with the King Abdullah University in Science and Technology in Saudi Arabia, and with the Florida State University. And with this, I would like to thank you for having shown up here, despite the fact that it's not a good moment. I need to express again my gratitude to the Nomis Foundation and to the EPFL as well. Thank you very much. If there are questions, I'm happy to take them.
Merci Tom. On va prendre quelques questions. As you said earlier, Fragen, questions, questions, use the language you like. Monsieur Ouvertes.
Bon, tout d'abord, merci pour cette présentation qui, pour moi, est un champ nouveau et j'ai l'impression que vous ouvrez de nouveaux champs, donc c'est très, très excitant. Par rapport à mon expérience personnelle, en fait, j'ai vu quelques années en Amérique du Sud où il y avait des projets de barrages sur des rivières qui descendent des Andes et se jettent dans le Pacifique, des méga barrages qui finalement n'ont pas été faits. Mais une question importante qui m'intéressait à l'époque, c'est quel est l'impact donc de ces grands barrages sur les rivières elles-mêmes. Et j'ai entendu parler de ce phénomène de clean river, ou clean water en aval de ces barrages. Ce n'est pas directement ce que vous mentionnez là, mais c'est peut-être un thème connexe. Je voulais savoir si vous avez par exemple fait ce genre d'analyse, par exemple en amont et en aval de ce genre de barrage, et si d'autres équipes travaillent sur ce sujet, mais quel est votre input sur ce sujet?
Ça va si je réponds en anglais?
Absolutely.
Okay,
No problem.
thank you very much. Okay, so thank you for the question, quite an important one. So in my lab, we've not yet been working on this topic. However, quite a bit is known on this. So whenever you stop the flow of the water, you change the entire ecosystem, not only in the Andes. I mean, you know, we don't have to go far, right? So here are two major points, or three. Okay, so we all rely on hydropower, that's clear. But those people that kind of lobby hydropower, should they never ever again tell us that this is like it's a renewable energy, but it's not a carbon neutral energy. Many of these reservoirs, many of them, they are major sources of methane to the atmosphere. Major sources of methane to the atmosphere. I repeat, major sources of methane to the atmosphere. I hope that you got the message, right? Methane is a very important, very potent greenhouse gas. Stop here. Then the third point actually is that, okay, so by breaking the continuum, right, today we understand rivers as, that's why I showed you networks, right? So, and there's like in the US, they say, we all live downstream. Whatever happens upstream, the signal is propagating or not downstream, right? So we do affect a lot the hydrology, of course, the biogeochemistry, ecology, the functioning of the systems, the water quality, etc., etc., etc. just have a look at the Rhone River. The bed of the Rhone River is completely armored, or it's what we call clogged, la colmation. It's a major problem, and that's because we have all the lovely reservoirs upriver, right? So it's a major problem because in a kind of a healthy river ecosystem, the surface water and the subsurface water communicate. We call this, it's one single water resource. Now there are kind of reflections to kind of declog the Rhone River. Some people think, oh, it's not a good idea because the groundwater in Valais is contaminated. We know that. It's contaminated. As you declog the bottom of the river, there's surface water entering into the subsurface, and it's pushing up the groundwater, you know, into an orchard, into the garden of a politician. I mean, these are the jokes in Valais, right? So you're literally pushing up the contaminated water, and all this ultimately because of the reservoirs, right? So there are many, many impacts, many.
Merci. Une autre question dans la salle? Ne soyez pas timide. In English as well, if you have any question.
Kids, any questions?
Yeah.
So I hope that the next time, whenever you go to a stream or river, right, in the mountains, please do me a favor. You take a couple of stones, right, and you're gonna see with your fingers, it's slimy. And this is the slimy megacity, right? These are microbes. You can even smell it. It has a very lovely smell. I'm not kidding, right? It's a very fresh smell. And there are millions and millions of cells on that very stone, producing oxygen, for instance, if there are algae that we respire. So I hope with this that the next time, whenever you go to the Alps and you see a stream, that you see it kind of with different eyes. That would be cool. Thank you.
Moi, j'ai une question quand même. Vous avez dit quand ça sèche, on ne sait pas si la vie reprend après.
Yeah.
C'est quelque chose qui est étudié chez vous ou ailleurs?
Non, c'est pas encore étudié chez nous, not yet, but I know, like for instance, in Antarctica, right, where this kind of season, where the water is fluid, is way shorter than, you know, in like here in the Alps, for instance, there these mats or these biofilms, right, they can become alive again within 2 days, and then they are freeze drying again. But I do suspect that it's similar here as well. Yeah.
Okay. Merci. Merci beaucoup.
Merci beaucoup.